Well, I kept the topic to work on tRNA because the RNA world is large, and now you have plenty of RNA to analyze with a big concentration on the non-coding part. But in terms of structure-function relationships, one of the oldest and really at the origin of all our structural knowledge about RNA was transfer RNA. It's the first non-coding RNA which has been analyzed and very important. And it is the three-dimensional structure of transfer RNA was done in '74 by crystallography. So it's about 40 years. And it took 20 years to get a second structure of another RNA, which was a small RNA, a ribozyme. Understand? And now things you realize are accelerating. Size, diversity problems, and so the club is large. But you will see that I hope I will convey the message that transfer RNA is still very new. You could see that already from Marat's talk and Goulad's, that we don't know everything about so functional transfer RNA, because what is important also is to link structure and function. So in the next half an hour there, I promise I won't extend too much for you. I will analyze just a few points. You know, I was told last night that I had to speak today, so at 11. So I have some excuse if the order is not correct. So just as an introduction, you know, you can have a classical hierarchy. That's the information that everybody has. You know, now, biocomputer, you don't have to read that because you don't need. You have computers that do that much better and longer. But that's the information. I have first one. With that, you go very little structural information. But you can see mutation, analogy, comparison. You enter in the world of structure-function relationship by really going to the 2D representation. And that was already a big step forward. You remember Marat showed you this first talk how the ribosome, the first achievement was to have this secondary structure by knowing just few basic rules. And so that's a very, very important step, but you reach really the structure-function relationship level when you enter in the third hierarchy, the tertiary structure. Here you have the tertiary structure, and you see here there are some spots which are highlighted. So the tertiary structure is not, it's made of double helical stems, that's classical, but more importantly are the deviation from that. And you have to imagine that the first successful model, which was predicted before the knowledge of the structure, was that of an intron by Eric Westhoff and François, okay, I forgot the name, both. By the way, the 2-3 slides I take is from a review from Eric and his student Cruz. They made that three-dimensional, a real three-dimensional structure. We happened to be, I say happened, yes, because that was the first successful model, a complex molecule. When the X-ray structure came out, the basic fold, everything was there. It's not just the fold, it's the interaction of things. But you have to know that this model was correctly built using the fragments. You know, it was just putting together fragments. So knowing the interaction, a lot of information, but the structural fragments were coming from the transfer RNA structure. So the transfer RNA contained all the block which could be used to make an intron, the deviation. Just to give you how things is exactly something similar to proteins when you have very complex things, but at the end turns with beta sheets, turn, loop, and helices and things, and then you can build. So the difficulty is to build, of course, and you have complex model. So here. So the model was there, but then structure came out. Here you have a three-dimensional model of an intron, group 2 introns, self-splicing. You see that when you look at that, if you don't have that type of analysis and you know who is where in terms of contact, it would be very difficult to analyze. That's why the modeling, computing, and things are very important, and of course the biochemical information. And all of, I'm just talking of in vitro things. In the third aspect, I will tackle very briefly at the end, the functional consequence, correlation. That's another. But having had that and all the contact, you can see what, if you do a mutation here and such a loop and that, how it does affect on a long distance interaction here. You know, these things which are long distances. No, here is from here. Or this part here. That you would not predict without a three-dimensional, and then your mutation effects. So that was the intron. The hammerhead ribosome, I told you that was a second crystal structure. And here you have the crystal structure. Here you have the secondary structure. And that's the easiest one can think of. You see, 3 stems to put together. You will see very trivial. I mean, everybody can do that, just push. That's a model, and then these are the two. I mean, there were, I don't know if you remind that, Mike, but there must have been dozens and dozens of structural models. These are the two most reliable, which fits all the data. But when you superposed these, that's okay. This one you can see, that's very, very far away. When you have things like that, differences, you can ask also another thing, which is very important. You should never forget, if you get one picture out of this prediction, and you predicted different things, so when you analyze, maybe you don't have all the interpretation, don't forget that there may be another conformation with another environment. That's a conformational adaptability. I will come to that. The adaptability, flexibility of these molecules and adapt. That's why it is so important to have, and Mart alluded to that when he was explaining to you how to analyze the interaction within the ribosome. You know, there can be different conformers. So it may not be that this one is irrelevant, but it's certainly not a major component. Okay, after this short introduction about the interest of bioinformatic analysis of structural information, come back to our friend transfer RNA. So, you know, this time people got a Nobel Prize just to have that cloverleaf. You know, other times. You saw the complex structures that we have today, and that's the representation. So you go from 1D, 2D, that's a 3D structure, one side and one side. What is amazing, I mean, you have this correlation. This one, we had that picture that was in '74. You know, everything fall in place. That was already quite significant in terms of information contact. But you know, but going from there to there, to know that these and these were in contact here at the corner. They have the distance here between the anticodon where the message is translated, and that's where the message is attached. And all the position of different things. We'll come through in some examples to see the relation between that structure and the extreme variety of function that this molecule carries. I will not tackle one problem which is also important, which are the modification of base. Here I'm talking just at the naked transfer RNA, sequence. Of course, a significant part of the life of transfer RNA is the modification. Some bases are modified things. I will not touch on that, although they are important. But the first question, having that structure in 1974, said that the challenge was to know how is the translation of the genetic code operating, the transfer of the information. We had the transfer molecule, there were protein doing the work, so this transfer of information was a challenge. It took 15 years before we got the answer at the three-dimensional level. So we jumped here. Here I have one example, which is the classic. I'm not going to talk about the synthetases, but this is the one we worked on, which is a so-called class 2, first example of class 2 synthetase, where you have the two transfer RNA and the protein. And you see how the CCA goes on. And so the interactions, here the active site, very charged, and here the anticodon. There is a communication, by the way. So this molecule is capable to transfer the information and to play in an allosteric way the regulation. Because what is important for us is to have the accuracy, high accuracy, and also the regularity in transcription. You don't want things to be completely, otherwise you want to process the genetic code, the protein, at a reasonable level. So regulation is very important. And so not only charging the reaction, but also to charge the proper transfer RNA with the proper message. That was a challenge. So here you have an example of the distance, and see, so the recognition here is important for that, but you see here you come to the classical clover leaf. Not everything, the structure is important, the global structure, but not every nucleotide is important for recognition, for function. So they are so-called identity elements, the ones which are important for the identity of the transfer RNA. That is, the identity is referring to the message, which means in order to be recognized, there are some key position. And these are variable. They can change from one transfer RNA to another. The fact that this was important was demonstrated by John Abelson's group. I remember it was in the 80s, something, when he just took two transfer RNA which were different, see, and he mutated one of them with the identity determinant of the other. Everything else was kept. He just transferred the identity element. And then the tRNA could be charged by the synthetase which was corresponding to the identity element and not to what the transfer RNA was. That was a clear demonstration, functional characteristic of the importance of these identity elements. And here you see, out of these 75 or so nucleotides, you have 3 important, major, and then minor. I will not enter the detail, but you will see that these major are making direct interactions. So you have interaction at the anticodon level, and you have a very important interaction, the CCN, with this base space. For this case, I will analyze just 2 cases. This one, just to show you what, and the other extreme of the family of synthetases, where one single nucleotide is being the identity element. So you imagine just one position is crucial to discriminate all other transfer RNAs, the recognition. Now that's a good example of RNAs, because we have a small, we don't have a large RNA and things, and then 75 still not a minor. So it's a good example we're talking about. Here we go to we continue with this transfer RNA aspartic, it's 2, 3. So here is the anticodon interaction. Here we concentrate on the base, the RNA part, not the protein. Of course, the protein are important, I'm not saying that, but you see that the stacking here is not perfect. And it's changed. Then you have a typical protein interaction. Then you have a conformation. Then the conformation becomes important. And everything. Here you have a modified base in natural concentrate, but every base here is making precise interaction. And especially the two identity elements which are here. By the way, this one is not a key, is not an identity element, but is part of the anticodon. On the other side, here, this base is making an interaction, very important interaction, but I just want to point out, because we'll come back to that later, in this stem, the fourth place, the G4 U69 is a GU. That's the electron density of the whole time. So it's good, but you can realize, I mean, it's you have seen before, especially with GU on my arm, it is quite significant. You see the wobble base in that stem. Why do I insist? You will see very quickly. So there is a GU, but it's not an identity determinant. It's in the stem. The stem is slightly affected, but nothing. It's not an identity, which means that if you mutate that GU, you make a GC or a U, nothing serious happens. You may play a bit with the kinetic, that's all. And here just to show that the GU, that's a 85 map, 99, you see, when you have high resolution, just that's what the GU looks like at high resolution with the water molecule, everything bridges. So that with crystallography, you're on the safe side to analyze the conformation. I can only confirm what Marat told you before. And with this system, we have a lot of crystal structure available in different states. So we can analyze another feature of interest, which is the dynamics of the binding. You saw the transfer RNA. I told you it's more or less rigid, it's like that, everybody. And then the protein. For this time on, we don't care about the dynamic of the protein. There is also a dynamic on the other side. You just look at the transfer RNA, the RNA. And so this is a summary resulting from lots of different crystal structure in different conditions. So we can design the binding motif. So the first binding occurs at the anticodon level. You have that. Then there is a step 2 interlines all along to bring the CCA end in the active site. So we have here. And that is the resulting final thing. But you see, in order to do that, of course, you have to recognize different key elements, but you have also to adapt. So there is another point that I would like to stress is the flexibility. Flexibility doesn't mean that you have to disrupt all these things. You know, between this molecule and this one, of course, that is highly mobile. Otherwise, the differences are not huge. That's significant. But I should stress to you also one thing. 1 or 2 angstroms movement in terms of energy, it can be few kilocalories. And 1 kilocalorie losing is 1 order of magnitude in terms of affinity. Just to, you have to imagine when we are playing here, the things you're saying with in vivo experiment, mutation that change, so you can modulate interaction very strongly. This is one aspect, for example, that could have been also described, but it could, for lack of time, on the ribosome recognition, you know. But I use that. Sorry. I couldn't resist that. An old map that you, I mean, old, a few years ago, map of the ribosome. Now he has much better, nicer. So this is good for me. That's the density of the transfer RNA on the ribosome. Just extract the density, of course. That's what you saw before. That's the cognate binding, near cognate, something which binds, but it's not good. You see, there is a flexibility that shows, that illustrates the flexibility of the transfer RNA, the importance of the energy. This one is moving. You can understand that this, if this one is like that, it is properly positioned, it will go in the right position. If it is more shaky, even if it can go there, it will be once every 100 times drops. So, which means there is a energy, kinetic, structure and flexibility, it's all tied together. There is another fact, but then I will not go in, just to describe you the distribution of electrostatic charges. By mutating at the protein level, you can affect the binding, mostly the backbone, the charge, and this is a long distance interaction and also a fine-tune in order to pre-orient. So the protein pre-orient the transfer RNA, binds with the anticodon, and then it goes in. So this is chaining, and both from the energy side and the kinetic. I told you I'm going to the other example again because I don't want to extend too much your patience. I showed you one example, aspartic acid with 3 major identity element. This one is an interesting synthetase, alanyl tRNA synthetase with tRNA alanine. tRNA alanine has one identity determinant, one base pair in the stem, a GU base pair in the 3rd position of the acceptor stem. And that was a bit mysterious, and maybe that is one of the points which was most analyzed in terms of recognition and discussed. I didn't have time to retrieve. I could have retrieved just to give you an idea of how science can move. And sometime I told you yesterday, I guess, or one day, that it's not because it's in nature that it is correct. You have to keep an eye. To explain that, there were a lot of papers. A GU, you see, is that. So the consequence of the GU, people were analyzing everything you can imagine. Now they can, so what I was alluding to was a famous Nature paper well, I don't remember the year, maybe you remember. I mean, actually, I discovered that they just opened a newspaper in France, Le Monde. Le Monde is a very important newspaper. On the front page, second genetic code is found. That was signed by a Nobel Prize, de Duve. Even found the second genetic code. That's not a small discovery, you must. That was because of the decoding of the transfer RNA. This is the decoding of the genetic code. That is the key step. And the idea was that there was a recognition by the minor groove important. Which is, that was correct. That it was important. What was wrong, there was no second genetic code. You have not missed anything. Your textbook is still all right. And that was, I guess, I don't know, 80 or something, in the later 80s. And 2 years ago, in Nature, I must say, not everything is wrong in Nature, the solution came. There are a lot, you would agree with me. Yeah, I have nothing against Nature, it's my reference for you. Actually, if you look at I have some paper in Nature. And so here is the recognition. We'll see now how it works, how with one specific position you can discriminate. We just zoom. You see here is the stem. What you see here, the anticodon is not involved in the recognition. And there is no identity determinant, so the notion of the identity determinant is linked to the recognition by the synthetase of protein which does the transfer. It's not. So here you have the contacts, the position, and here you have the active site. View down, you see here how the acceptor stem goes. There is a protein. The active site is there at the end. So you have a stem goes right in the active site. I zoom. That's a bit more messy, but then just if you compare the transfer RNA, the two that we just analyzed, I mean, we could superpose the structure of the bound tRNA, not the free tRNA. So we just took the two tRNAs, they're both bound to their cognates in the case. You see they are very similar, very close. The main difference has been in there, but you see, no huge difference. So we are really looking at the atomic level details here. You remember for one, the recognition was there and there, anchoring. For this one, the key residue is there. What happens? What is the solution? I told you there were maybe 100 papers working on the modification of tRNA, trying to explain how the minor groove was accessible. Because one thing, if you look at the structure of the stem, RNA stem, it's not accessible in the major groove, except at the end when it's finished. Only the minor groove. If people look at just canonical stem, you see in DNA, the bDNA, major groove is accessible. The minor groove has absolutely no specificity. So you can read the basis and turn or not. With RNA, that's not true. That's why people were concentrating. Tend to forget actually that at the end of the stem, of course, there is an accessibility. Okay. And here is a close-up. So the density is not the greatest in the world, but that's the base pair we are concentrated with, GU. And here is something interesting. It's a protein. It's just a slice in the stem. That's protein. So at the end of the stem, it is an opening, and then the protein can reach the major groove, which people had discarded as a protein. That's why it was so difficult to find a molecular mechanism. And here you see normal, that's the interpretation. That's the helix with the GU, you see, with the wobble pair, the G1 C72, the C4 GC, everything is correct. So the only point, the critical point is the G3U70, where we have the wobble pair, but what makes the difference now is we have the protein which is reading a GU base pair. Then the protein can distinguish between the wobble and non-wobble. And in order to prove that, people did a mutation, very classical, that had been done hundreds of times. A3U. You restore, you make a standard base pair. But this time they could look at the density. That's where crystallography comes into. Told you, seeing is believing, this time you can. Look, the density, the G3U, is a decoding of the pair. When you do the mutation, the protein has shifted a little bit, I think. There is no reading here. So there are two effects of the GU. First, the decoding, and then the stacking. I will not extend. When you have a GU and over regular basis, the stacking is affected. So if you control the position there, you stabilize the rest. And if the conformation is important, that's my point. Conformation is important. When you have the right conformer, that is the proper sequence with the proper stacking, you block in the right position. It's a finely tuned motor. Then the rest follows and goes in the active side and charge. There is a neck here, bottleneck. If you have a mutation, you have a slight distortion. You don't need a lot. The wrong stacking, it's a different stacking. All you need is 2 or 3 angstroms, not even. I think it's 1 angstrom. And then instead of going straight, you bump to the neck, you're out. So you see, that's the mechanism of selection. And an amazing thing is that one base pair was kept. These are among the most ancient proteins in terms of decoding and we know we are talking about mutants and things. Most ancient. with one GU base pair as an identity determinant is kept sufficient to discriminate, and if you mutate that in your body, you are dead, I can assure you. But fortunately enough, that doesn't happen. And I think this is a good example of diversity. You have seen before the importance of the pairing. There is a cost behind, of course. I'm giving you the steric consequence. There is an energy cost behind, the play. Always few angstroms. You can always force. Not in this case, but if you force, you need energy. And then it's unstable. It won't stay. Other consequences. I think I told you I would not bother you more than half an hour, and that's all I could do. Thank you.